Okay, decomposition, probably my favorite topic. I have a lot to say about decomposition. So first of all, to study decomposition, we first collect leaves, sometimes from specific individuals, sometimes from whole communities. We dry the leaves, we weigh them out and place them into mesh bags, and we provide little unique tags so we can identify the bag and measure its weight before and after we expose it to the environment. Then we transport the bags to the field, we incubate them in a stream or a lake, and then we pull out subsets of bags over time and transport them back to the lab for processing. And then we dry and weigh the leaves again. And then we often combust the leaves at the end to determine what's called ash-free dry mass. So here are just some close-up pictures of a few different types of leaf litter bags. Sometimes you take punches of leaves if you want to maintain kind of the same surface area, and then you can see little ID tags in there, and these color-coded cable ties. The color-coded cable ties are really helpful when you're going back to the stream to collect your different harvests, because you can tell which litter bag is meant to come out at which point in time. So the decomposition process is the physical and chemical breakdown of detritus. And so there's first kind of a leaching stage where soluble materials are just leached out. Then there's a fragmentation stage where you're having microbial colonization and increases this, and then the microbes start breaking things down and fragmenting the leaf, which increases the surface area and causes little rifts in the leaf, allowing other things to colonize. Microbes can chemically alter the leaf, and then you have aquatic insects coming in and colonizing and continuing kind of the manual breakdown and conversion of the coarse particulate organic matter into fine particulate organic matter, feces and fragments. So leaching is this first step in this moving water-soluble compounds away from the decomposing material. And it can happen while the leaves are still on the plant, when there's rainfall, but it's most important early in the wetting process when the leaves first get wet. We lose things like nutrients, and then more labile things like sugars, amino acids, and proteins. And this is the kind of the first phase. And you can have a really rapid loss of mass in the leaching phase. Some leaves, when you first put them in the stream, will lose up to 25% of their mass just in the first day or two, due just to leaching. And then you're left with these more recalcitrant things like cellulose and hemicellulose and lignin in these later stages of decomposition. So fragmentation, we have fresh litter, which is typically protected from attack by intact things like waxy cuticles. But over time, and things like bark or skin or the exterior waxy barriers on leaves are protecting things. And then plant cells are protected by lignin and cell walls. So it takes time to break all of these things down. Fragmentation, maybe it's physical fragmentation, rocks bouncing around with the leaves, or the leaves getting wrapped around something and kind of ripped, increases surface area then for microbial attack. And fragmentation is important in both aquatic and terrestrial ecosystems. And then once you have kind of cracks and fissures in the waxy cuticle layer and chunks taken out of things, and you can have fungi and bacteria start breaking things down. And so the fungi and bacteria colonize, and then they start altering the leaf litter chemically. So some of these organisms use what are called exoenzymes. So they release enzymes into the environment outside of themselves, which can break down these recalcitrant compounds and turn it into CO2 and nutrients. And then leaving behind some of the more recalcitrant compounds, or forming new complex, even more recalcitrant compounds. So here's some pictures of just some sporulating fungi on a pine needle underwater. And the fungi are the main initial detritivores. They really reach their peak at about 4 weeks after the leaves hit the water. This is a giant lignin molecule. And the fungi access nutrients through their hyphae, and they have a competitive advantage over bacteria in the early parts of the decomposition process. And they in particular use exoenzymes to break down this giant molecule, which is called lignin. And the white rot fungi are particularly good at this. So here we see a white rot fungi breaking down woody material. And they use a lignin peroxidase enzyme to basically explode the lignin molecule extracellularly. So it's a really cool enzyme. So if we can see, if we plot mass remaining, this ash-free dry mass remaining through time and stream, we can see that early leaching signature, and then we can see this kind of more steady decomposition rate of those recalcitrant materials through time. So it usually looks like an exponential decay curve. And so what we can do is take the natural log of that data, which then shifts the values to show a straight line. So the slope of this straight line then is the exponential decay constant. And if I go back here, you can see the equation for exponential decay, where we have the mass at any time t is equal to the original mass or the initial mass m sub 0 times e, which is the mathematical number e, to the negative kt, where t is time and k is the decomposition rate constant. And the cool thing about decomposition is we don't actually have to solve for this equation, but if we convert the mass remaining data to the natural log and we do a linear regression, then the slope of this line then becomes k, which is the decomposition rate. So it's really cool because we can calculate the decomposition rate using linear regression. And the decomposition rate in this time is the mass lost per day. Okay, and so what are some of the factors that control decomposition? So many. The physical environment, the type and quantity of the substrate, and the characteristics of the microbial community all can control decomposition. So things like the surface area of the leaf, stream conditions like nutrient concentrations, the types of invertebrates and microbes present for colonization, concentrations of lignins and tannins in the leaf itself, carbon to nitrogen ratios, other nutrients, phosphorus, and then more surprisingly, things like the genetics of the leaf or herbivores in the terrestrial environment, or things like CO2 exposure in the terrestrial environment or exposure to pollutants like ozone and things like that. So there's lots of ways that streams can influence forests. So streams can export insects to forests. They can provide habitat for larval stages of insects. They can provide water to forests, groundwater recharge. They transport materials out of forests and process their waste. And then we'll learn that they can help process nutrients through nutrient spiraling. There are cool things that terrestrial environments can do to influence streams as well. So forest floor runoff, they provide the habitat for adult stages of aquatic insects. They can provide inputs of terrestrial insects to streams feeding fish at certain times of year. Shading and cooling, exotic species invasions can have really strong effects on aquatic systems. Evapotranspiration takes water out of the stream. And then things like woody debris and leaf litter inputs come into the stream. So I'm going to focus mainly on leaf litter inputs. It's the area that I've done the most research in myself. And some of the questions that I've asked are, how do different species of trees influence leaf litter decomposition in streams? How do different mixtures of species influence streams? Do they have additive or emergent effects when you mix leaves together? What kinds of alterations to leaf litter by terrestrial agents, things like herbivores, pathogens, endophytes, how can those things influence leaf litter? Environmental stressors, things like drought, pollution, infection, And what about the genetic makeup of the leaf litter? So these are all things that I've studied both here at Evergreen with students and previous to that in my PhD research. And so here's a bunch of just papers that we've published on leaf litter decomposition. So things like, can leaf litter from genetically modified trees affect aquatic ecosystems? How do salmon carcasses interact with leaf litter to influence decomposition? How do fungal endophytes influence decomposition? How does the plant's genetics affect decomposition rates? So when you look across broad plant families, you can see that there's a lot of variation in their decomposition rates. This is from Webster and Benfield in 1986. But if you look within a plant species, you can see equally large variation in decomposition rates. So here's a paper where we were looking within 2 different species of cottonwoods and their hybrids and comparing it to family-wide variation in decomposition across plant species.